Dear colleagues, let me start my first lecture in English for you in the second semester. I will speak about urinary system today, and let's start immediately with the first and the most important organ of this system, which is a kidney. Kidney is a paired organ situated in the retroperitoneal space. It is surrounded together with the adrenal gland by adipose capsule, adipose tissue, which isn't firmly connected with the surface of a kidney, so it isn't usually present on our slides. The first layer which you can find usually on the slide is a fibrous capsule. Fibrous capsule, which you can see for example here, is the first layer you'll find on the slide. Macroscopically, the kidney is composed of several parts. You will see a cortex, you will see a medulla, and you will see a renal sinus. Renal sinus is a space which is filled with loose connective tissue and which contains many structures. It contains the beginning of urinary passages, it contains vessels, it contains nerves which accompany these vessels, so it's quite interesting area in the slide. Let's start with microscopy, with microscopic structure. And the first layer, which is really on the surface of many slides, is a capsule, fibrous capsule. You can see that it's quite thin, it's a high magnification here. Its superficial layer is composed of dense, irregular connective tissue. Its deeper layer is composed of loose connective tissue here, and in between there are some vessels which are supplying the tissue. And immediately under the capsule is a renal cortex. So this tissue here is renal cortex. You recognize renal cortex and renal medulla even in the macroscopic slide. You will see surely in the anatomical department this lighter tissue which is granular, which seems to be granular structure. If you look at it carefully, this is the cortex. You can see that the cortex isn't only under the capsule, but it penetrates to the deepest part of the kidney and is in the contact with the renal sinus. This space is a renal sinus. These extensions of the cortex penetrating deeply to the kidney interior are called renal columns. Renal columns, columnae renales of Bertini. Renal column. So this is one renal column. These darker areas, usually triangular in shape, are so-called renal pyramids. Pyramids, which is a good name corresponding with their shape, and they are composed of medullary tissue. So this is a medulla. So you can see that the medulla of a kidney isn't continuous. It is divided into the several pyramids. Each pyramid, together with the corresponding part of the cortex, forms renal lobes. So the kidney is composed of lobes. Nevertheless, they aren't usually visible on the surface. Only in small children, the kidneys are so-called ranculinized. That means they are deep grooves between lobes, but in adult kidney these grooves become shallow or disappear completely. So the kidney is composed of lobes, and each lobe is also composed of lobules, but it will be visible only in the microscopic level, not in this macroscopic level. And when we look at the sinus, here arteries are visible. This is a branching of renal artery, which are specially dissectioned in this specimen. Nevertheless, in the same area you can find veins, and you can find renal calyces and renal pelvis. That means the beginning of the urinary passages. Each renal pyramid is situated in the minor calyx, and this area of the renal pyramid, this terminal part of the renal pyramid, is called renal papilla. Renal papilla, that is why here is this letter P. So there are also renal papillae. Okay, so let's go to microscopy. This is a middle magnification of the border between the cortex and medulla. How to quickly recognize cortex and medulla. You can see that both or all, the whole slide here contains many tubules which are in cross section. That means their shape is spherical, but they can be also longitudinal sections. That means there are long profiles of these tubules, and these tubules are everywhere. They are here up and here down as well, but here up, let's say above approximately this level, is the something else among these tubules. There are so-called renal corpuscles. This is one renal corpuscle. This is the second renal corpuscle. Together I can see here 1, 2, 3, 4, 5, 6, 7, 8 renal corpuscles. And this is the easiest way how to recognize the renal cortex. Renal cortex contains renal corpuscles. So this upper half of this picture is the renal cortex. Renal medulla doesn't contain any corpuscles, so this lower half of the picture is a renal medulla. The tubules of the cortex and medulla also differ. You can see it. Here down, tubules are usually smaller than up. I will speak about all renal tubules in detail, so I'll describe all the differences. But the easiest way how to quickly recognize, even in the small magnification or even in the macroscopic picture, the renal cortex, is the presence of renal corpuscles. Renal corpuscles are big enough to be visible as small grains in the macroscopic specimen, and that's the reason of the granular appearance of the cortex. This is a small magnification, and so we can see more from the border between cortex and medulla, and you can see that this border isn't straight like it seemed to be in the previous high magnified picture. The border between cortex and medulla is highly curved. There are long processes or protrusions of the medulla going deeply to the cortex, going almost to the surface of a cortex. The distance between the tip of this medullary protrusion and the surface is only very small. Here is a measuring bar, so 1000 micrometers, that means 1 millimeter is here between. So the medulla penetrates deeply to the cortex. These projections of the medullary tissue going to the cortex are called medullary rays. Medullary rays, or if you like more Latin than English, striae medullares. Striae medullares, medullary rays. And their arrangement is dependent on the arrangement of renal vessels. Renal vessels are going every time between the medullary rays. Mostly arteries are going here, they are branching then towards the renal corpuscle. There will be a picture of the renal vascular supply, so I'll explain it better. And the medullary rays divide the cortex of the kidney into lobules. So each lobule of the kidney is one medullary ray together with surrounding cortex. Yeah, I didn't choose a good example. Sorry, I delete or these lines here, and I'll try to draw one lobule of kidney. So border of lobule is here, of course here on the surface, and let's say somewhere here. So lobule of a kidney is a medullary ray in the center and all surrounding and covering cortex. So there were lobes, which are macroscopic structures, and lobules, which are microscopic. And again, you can see even in this small magnification that the best and the biggest difference of kidney, medulla and cortex is the presence of corpuscles in the cortex. Renal corpuscle is here, 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 or here, here. So all this tissue where corpuscles are present is a cortex. Yes, and here is once more how to define the renal lobule. So lobule is medullary ray in the center, it is this, and surrounding cortex. And you can see that there are arteries at the border of lobules. These arteries are logically called interlobular arteries. So interlobular arteries are between lobules. There are also interlobar arteries and veins, which are situated between pyramids in the tissue of renal columns. Yes, and here is a high magnification of one lobule. In fact, I don't know if I can find some interlobular artery. Maybe here. Here is a small artery or arteriole situated in a good position. So I think it can be a section of renal interlobular artery. So here or here as well. Yeah, this. So you can say that this is the border of a lobule, and on the other side approximately here can be the border. Here I don't see this arteriole and medullary ray containing tissue of the medulla in the center of a lobule. Histologic composition of a kidney is quite complicated, so I'll divide it into three parts. I will speak about the vascular component of the kidney, which is very important because, as you probably know, the main function of a kidney is filtration of blood, clearing of blood. So the blood supply of a kidney must be very special. The special thing is that there is arterial portal system. That means the system of two capillary networks connected by an arteriole, which can be called portal arteriole, but in fact we call it afferent arteriole. These two capillary networks are composed of different types of capillaries. Glomerular capillary network, situated in each renal corpuscle, is composed of capillaries with pores. So-called peritubular capillary network, which is the second network, connected with the glomerulus by an afferent arteriole, is composed of fenestrated capillaries. That means of capillaries having diaphragms in their fenestrations. And there are also vasa recta. I'll explain it. They are special vessels of a medulla. Of course, there are also veins. The second histologic component of a kidney are renal tubules. There are many tubules, and they are from functional and also developmental, embryological point of view, divided into two groups of tubules. We can distinguish tubules of nephron. Nephron is the main functional and structural unit of a kidney, and it contains several tubules which have one embryological origin. They originate in so-called metanephric blasteme, but it's time you will hear the special lecture about the development of kidneys, so let's wait for this. The second group of renal tubules are collecting tubules, which are connected with tubules of nephron. Nevertheless, they are developing from another source. They are developing from so-called urethral bud. And the third component of kidney is a renal interstitium, which means the tissue situated everywhere between tubules and vessels. The main tissue here is a loose connective, but it contains some special cells, so-called interstitial cells, mesangial cells. They belong to this interstitium, but not normally into loose connective tissue. Several words about the vascular supply, about the vascular component of the kidney. Kidney is supplied by a renal artery, which branches into main segmental arteries, and they then go in the renal columns in between renal pyramids as interlobar arteries. Interlobar arteries go to the border between cortex and medulla. They branch and they go parallelly with the surface as so-called arcuate arteries, and they send branches to the cortex, and these branches are interlobular arteries. You already know. You know that they are situated in the cortex at the border of renal lobules. Interlobular arteries branch. They branch in right angles, and their branches are called afferent arterioles. Afferent arterioles, you will see them in this higher magnification of the schema. Afferent arterioles are finished by the first capillary network in the renal corpuscles. This capillary network is composed of capillaries with pores, and these capillaries are called glomerular capillaries, and they together form a renal glomerulus, one of the parts of renal corpuscle. These capillaries join again together, but their vessel collecting blood from capillaries is not vein or venule in this case, but again arteriole. That's why there is a portal system, and this second arteriole is called efferent arteriole, and the second efferent arteriole again branches into the capillary network. There is then peritubular network, peritubular capillaries, which are fenestrated, or if the glomerulus or renal corpuscle is situated near the border with the medulla, that means if this is so-called juxtamedullary nephron, some branches of the efferent arteriole can go to the medulla as straight vessels, so-called vasa recta, which accompany the special part of renal tubules, which is called loop of Henle. In both cases, blood is collected by the arcuate vein then, or there are also stellate veins and interlobular veins. All these veins join together to go to interlobar veins, and at the end to renal vein and to vena cava inferior. So that is the vascular supply of the kidney. Very important. Please, you must know it not only for histology but also for anatomy. So I hope you will know the vascular bed of a kidney. And now to the renal tubules, to the tubular part of the kidney. As I already said, some of these tubules belong to nephrons, and I also already said that the nephron is basic morphologic and functional unit of the kidney. So nephron is a really important thing. Nephron is possible to define as a system of tubules. The beginning of the nephron is Bowman's capsule. Bowman's capsule doesn't look like tubule, but it is in fact the blind end of the tubular system of the nephron. Bowman's capsule becomes during the development a part of renal corpuscle, has got its parietal and visceral layer or leaf, and is very important at the beginning of urine formation because it has a filtration function, and it participates in the formation of primary urine. Primary urine is produced in renal corpuscle. Each kidney has got about 1 million of renal corpuscles, and so it isn't surprising that so much urine is formed every day, about 180 liters per day. The primary urine then goes from Bowman's capsule to another parts of the nephron. The first of them is proximal tubule, then urine goes to Henle's loop or loop of Henle, and then to distal tubule. Each nephron has got these three parts and Bowman's capsule, so in fact together 4 parts. Each nephron has got Bowman's capsule, proximal tubule, Henle's loop, and distal tubule. Proximal tube, Henle's loop, and distal tubule are important for urine adjustment, for the reduction of its amount, of course, for filtration, back filtration of many substances which were filtered into the primary urine in the renal corpuscle. Here is a schema of a nephron. So, renal corpuscle contains a Bowman's capsule. So let's say this white or whitish area of the renal corpuscle is Bowman's capsule. You will see in a higher magnification that it has its parietal and visceral layer. Then urine goes to proximal tubule, which is blue here, to Henle's loop, which is orange, and to the distal tubule, which is this red part. And at this level, distal tubule is connected with the system of collecting tubules. Collecting tubules represent the parts of the second system of tubules, which have a developmentally different origin, and so I will describe them later. So now I am describing system of renal tubules of each nephron up to this point approximately. As I already said that each kidney has got 1 million renal corpuscles, and that means it contains also 1 million nephrons, because each renal corpuscle is a part of one nephron. So I'll describe now all parts of the nephron, and let's start in the renal corpuscle. Renal corpuscle or corpusculum renale is about 200 micrometers in diameter, so it is just at the level of naked eye visibility, but very small, and it is composed of 3 components. It is composed of its vascular component, which is called glomerulus, and which is composed, as I already said, from capillaries with pores. Then it has got its tubular component, which is called Bowman's capsule, having parietal and visceral layer, and then it has some amount of tissue between vessels and Bowman's capsule. There is some amount of tissue filling free spaces, and this is called mesangium or mesangial tissue. So it has vascular component, tubular component, and interstitial component, similarly as has the whole kidney. Let's start with the description of glomerulus. Glomerulus, which means the tuft of very special capillaries. We can see such capillaries here in the section. This is a lumen of a capillary. Capillaries of a glomerulus are branches of afferent arteriole. Afferent arteriole is this one. It isn't easy to recognize which is afferent arteriole and which is efferent arteriole. Usually efferent arteriole is smaller, but as you can see in this picture, there isn't any difference. But okay, the author of this picture has a label here, afferent arteriole, so why not? So all these capillaries are branches of this afferent arteriole, and when we look at them in electron microscope, not in scanning but in transmission electron microscope, we can see the endothelial cell. It is this. It is a nucleus of endothelial cell, and we can see its lateral processes of the endothelial cells, and pores. Pore is this hole. It isn't very high magnification, but I hope you'll believe me that these holes in the capillary wall are really free, that they are not bridged by any diaphragm or some membrane or something else. There is nothing, and so the only limitation for the substances passing through these pores is their size. The size of pores is approximately 70 to 80 nanometers. So smaller structures can pass through the pores, larger structures no. So this is the lumen of glomerular capillary, and the glomerulus in the kidney is the only place in the body where the special capillaries with pores are situated. These capillaries with pores are described in the Junckera's textbooks as capillaries with fenestrations without diaphragm. It doesn't mean anything else, but I think that the term pore is simpler and easier for students than fenestration without diaphragm. So please try to use it. Of course, if you say fenestration without diaphragm, it isn't any problem. I skipped several pictures, so I will go back. Schema of glomerulus. It is possible to see glomerulus in special specimen, where is the content of glomeruli filled with some colored material. Here is once more. You can see how glomeruli are situated near the interlobular artery. The interlobular artery is sending branches, afferent arterioles, and this is nice picture showing development of renal corpuscle. Let's imagine that there is at first arteriole. Arteriole is branching to form a tuft of capillaries. Near the tuft of capillaries is a blind end of nephron, just developing nephron during the embryonic development. These two structures meet, and the tuft of capillaries invaginates to the interior of this blind end of the nephron, and this blind end of the nephron then becomes Bowman's capsule. And that's why Bowman's capsule then contains its parietal layer, which is composed of simple squamous epithelium, and its visceral layer, which is covering all surfaces of all capillaries inside. And it is composed of very special epithelial cells. I will describe them in a moment. And this is the picture of scanning electron. From a scanning electron microscope, you can see a little bigger afferent arteriole, little smaller efferent arteriole, and tuft of capillaries between. It is special corrosive preparation where Bowman's capsule was removed, but normally it's everything surrounded normally by Bowman's capsule, of course. Okay. Bowman's capsule is composed, as I said, from visceral and parietal layer. Parietal layer is simple squamous epithelium. Visceral layer is composed of podocytes. Podocytes are epithelial cells, which are very special. It is difficult to speak about their shape. They are neither squamous nor cuboidal or columnar. It is impossible to say to explain their shape by one word. You can see that the podocyte has a body, and this body sends processes which are surrounding capillaries of the glomerulus. These are so-called primary processes. So body with a nucleus and some big organelles, then primary process, and primary processes don't touch the surface of capillaries properly. There is some distance between the primary process and capillary surface, and this distance is bridged by secondary processes of podocytes, which are called pedicels. These secondary processes are called pedicels. And they touch the surface of a capillary, or better is to say they touch the basal lamina of the visceral layer of the Bowman's capsule. So the podocytes are not in direct contact with their basal lamina. They contact the basal lamina only by their pedicels, small processes, small feet. So let's look at several pictures of these very special cells of the podocytes. This is a picture from scanning electron microscope, and we can see, for example, here a body of a podocyte. Here is another body of the podocyte. Here is also a body of the podocyte. These bodies send primary processes. One primary process, second process, third process. They look like octopus. If you have some ability to imaginate it, and these primary processes are not straight only, they are branching, and they are trying to cover. Here is one long primary process which is covering the outer surface of the capillary. This magnification of this picture is small only, but here is a square containing one podocyte and one loop of a capillary, which will be in the, I think I can use the this schema, which can be in this schema. So if you look at it once more, here is a body and some primary processes. Let's look at it. Here is a body. We are sure that there must be nucleus inside and some primary processes. Yeah, and this is a loop of a capillary, and we can see how this podocyte is covering the capillary by its primary process and by its pedicels or secondary processes. And you can see that a pedicel from one primary process is in the contact with pedicel from another primary process, and this another primary process can be a branch of another podocyte body. So there are places where pedicels of two podocytes come to the intercellular context, intercellular junctions. But intercellular junctions of pedicels are very special. You can see that there are some spaces, some slits between. These slits measure about 20 to 30 nanometers, so are very, very small, and they are bridged by something which can be also called diaphragm, something similar to the diaphragm you know from fenestrated capillaries. We know the composition of the diaphragm between pedicels. The main protein of this diaphragm is called nephrin, and there are also several another proteins. One of them is also podocalixin, which is special by the negative electric charge, and this is important for the transport of substances through this filtration slits. It serves as the ion slit or ion. These slits with podocalixin serve as the ion sieve. Okay, so these are podocytes and their processes at the level of electron scanning microscopy. This is the same but in the photograph. So body of podocyte, primary process, and secondary pedicels. This is also P, but pedicel and pedicel from another process, which can be a process of another primary process of another podocyte. And this is the level of transmission electron microscopy. Transmission electron microscopy can show something more, can show deeper structures, can show the contact of the podocyte and capillary. And what we can see at this level, we can see that the basal lamina of the podocyte, that means the basal lamina, or I will show it in this inset, in this higher magnification, basal lamina of the podocyte, which is this one, you recognize lamina densa, lamina lucida, is fusing here with the basal lamina of the glomerular endothelial cell. This is the lateral process of endothelial cells. These big arrows are in the pores where you don't see any diaphragm, and there is also lamina lucida and lamina densa, and these two laminae densae fuse completely. We don't see any gap between laminae densae of the podocyte and of the endothelial cell. This is the same situation as you know from the lungs, where is the contact of pneumocyte and endothelial cells also contain complexes of fused basal laminae, and we use the same terminology here. So this complex of fused basal laminae is composed of lamina lucida subendothelialis, it is this, so subendothelialis, lamina lucida subepithelialis is on the other side under the podocyte, and complex of fused laminae densae is in the center. How I recognize what is podocyte and what is endothelial cell are filtration slits. These smaller arrows show filtration slits which are smaller. It's the first thing. They are much smaller than pores of the endothelial cell. And the second thing is that they are bridged with diaphragm. We know that it contains nephrin, podocalixin, and other glycoproteins. So this is the place where blood is filtered. Let's imagine some molecule which is situated in the blood, which is on this side. This molecule must be smaller than I made. So let's say this is the molecule, and it goes through the pore. There is no problem to pass it because our molecule is very small. Then there is a complex of basal laminae, where are substances serving as a filter, mostly collagen type 4 in the lamina densa. This is mechanical filter, very important, and also heparan sulfate in the lamina lucida of both sides, which serves as one of ion filters, and another ion filter is podocalixene in the filtration slit. So our molecule, let's say it isn't catched by these filters, so it goes on, it goes through and penetrates to this space here, and is then here, and this is so-called urinary space. Urinary space where primary urine is formed. If I'll try to show this pathway in this smaller magnification, here is capillary lumen, so our molecule was here. It goes through the, not here, here is a too thick part of endothelial cell, but let's say here, it goes through the pore, through the complex of basal laminae, through filtration slit, and we have got it in the urinary space. This big structure is a body of podocyte. You can say that it is also in the urinary space, but it is connected with the surface of capillaries by its primary processes, which is here. And that is. These are, all these things here are pedicels. Pedicels. And we know that not all these pedicels have to be processes of this one podocyte. Some of them probably yes, but others are processes of another podocyte. We don't see its body in this section. I already said everything about this picture, so just to repeat it, a pathway of our molecule from capillary, which is at this side, pore, lamina lucida subendothelialis, fused basal laminae or fused lamina densae, second lamina lucida, diaphragm in the filtration slit, and here is a urinary space. And high magnification of the same, and I hope you now can recognize that the capillary lumen is here down. This is a lateral process of endothelial cell which is perforated by pores. So molecule goes through the pore, then it goes through the complex of all layers of fused basal laminae, through the filtration slit where is the diaphragm, and it is in urinary space. This is one pedicel of the podocyte. Here is another pedicel, another one, and you can see that these two are surely branches of this primary process, but probably this one is a branch of the primary process of another primary process. Yes, okay. We can visualize basal laminae in the renal corpuscle by some techniques. For example, this left picture is a PAS reaction. PAS reaction is good for recognition of the basal laminae because they are rich in glycosaminoglycans. So we can see that they are PAS positive. Each capillary is surrounded by basal lamina here, but we can see much thicker layer pus positive around the whole renal corpuscle, because under the parietal layer, parietal leaf of the renal corpuscle is a full basement membrane containing also reticular fibers, which are also PAS positive. And the second picture at the right side can show the same things stained with Alcian blue. It's very similar situation. Mesangium, which is found in the glomerulus and which is represented by mesangial cells and also by a mesangial matrix, is not only something filling free spaces, but it is also important for the phagocytosis of some bigger molecules which can accumulate in the filtration barrier, and there is necessary to clean this barrier. They have also some contracting abilities, so they contract to regulate the blood flow through the corpuscle, and they also secrete the mesangial matrix, which is composed of glycosaminoglycans, some proteoglycans, and which is filling all free spaces there and supports glomerular capillaries. So the mesangium is very important, mostly for the cleaning of the basal laminae in the renal corpuscle. High magnification showing some mesangial cell. Mesangial cell is this, mesangial cell is here. We can distinguish them from podocytes. Podocytes have got usually spherical nuclei, lighter cytoplasm. Mesangial cells are more basophilic. They're high magnification, mesangial cell, and some mesangial matrix here. This is podocyte. So I think it is possible to distinguish them. Okay, we will go to the next parts of renal tubules. So as I already said, the beginning of renal tubules was a Bauman's capsule, and then we'll go to proximal tubule. Proximal tubule is the longest part of the nephron. It is highly convoluted. It measures about 60 micrometers in diameter, so it is not only longest but also thickest part of the nephron's tubule. The urine then goes to short straight part of the proximal tubule, which is in fact equal to the thick portion of descending cliff of Henle's loop. Then the Henle's loop become much thinner, and this stands as a thin segment or thin limb of the Henle's loop. Then there is a different situation in two types of nephrons. I explain it, but usually we have also ascending part of the thin segment or thin limb of Henle's loop, and then urine goes to the thick ascending segment of Henle's loop, which is in fact equal to a straight part of distal tubule. Distal tubule has then its convoluted portion, which is shorter and thinner than the convoluted portion of proximal tubule. And for example, I don't know exactly where, but let's say at this level, distal tubule ends, and the urine goes to collecting tubule, which doesn't belong to nephron because it has different embryological origin. And then collecting tubules join to form collecting duct, and urine goes to urinary passages. And let's look at the proximal tubule in the microscopic picture. Proximal tubule, as I already said, is about 60 micrometers in diameter, so this distance is 60 micrometers, and it is lined with simple cuboidal epithelium. Cuboidal cells of the proximal tubule are very big, and so we can see usually only few nuclei in the section. We can see huge parts of the wall having no nucleus because the cells are so big that we can cut them laterally in the area where is their nucleus. So cuboidal cells of proximal tubules are big. They are connected by interdigitations. Their lateral walls are very, very complicated, and so we don't see borders between cells. We usually cannot see some lines, some borders between cells. We can see only spherical nuclei, which are situated in the center of the cells, and very, very highly eosinophilic cytoplasm. This eosinophilic cytoplasm is eosinophilic because of the content of very many mitochondria. The proof of so many mitochondria is these pictures down. It is special cytological staining of mitochondria. It's using of the Heidenhain's hematoxylin. And so you can see how many grains, where each grain represents some mitochondrion, are present there. Electron micrograph at the right side can show mitochondria as well. There are very many. And we can see another thing. We can see very many and very long microvilli on the surface. You already know from intestine what does it mean when the cell has got so many and so tightly arranged microvilli. This is a brush border. Brush border. Sometimes you can find also the term striated border, but much more frequent is brush border. So the proximal tubules are lined with brush border, and this is a reason why we don't see very well the lumen of proximal tubule. When I delete this drawing here, you can see that the lumen isn't cleared. It's filled with something. Sometimes it will look like fully obturated by something. It can help you when you try to recognize proximal tubule, and the reason of this are long microvilli. The cells of proximal tubule don't contain only mitochondria in their cytoplasm, they contain also many vesicles because they absorb a lot of substances by their brush border. You know that brush border is a specialization serving for absorption, and this is the case of proximal tubules. They absorb many substances, many molecules, proteins, polypeptides, carbohydrates, also a lot of ions, water. So they reduce very much the amount of primary urine. They don't only absorb these substances, but they also metabolize them directly. So they have got a lot of lysosomes where proteins, polypeptides and other macromolecules are digested, and they also have got very deep invaginations of basal surface where mitochondria are present in these folds, and we know that this situation where the basal surface of some cell is enfolded and very many mitochondria are in this area, you know that it is a so-called basal labyrinth. So the next typical feature of the cells of proximal tubules is a basal labyrinth. So they have brush border and they have basal labyrinth. They have both these specialization. One is the specialization of apical surface, second is specialization of the basal surface, and they have also many interdigitations, so also their lateral processes are very specifically specialized. They are underlined by basement membrane, of course, and under the basement membrane is a peritubular network of capillaries. These capillaries have got fenestrations with diaphragma, and they absorb all the substances which are transported through the cytoplasm of the cell, or which are metabolized here and then released to the capillaries at the basal site. High magnification showing all the features I said. Brush border almost filling the lumen, spherical nucleus. Here is something like striation on the basal surface. It is a proof of the basal labyrinth. We don't see lateral walls of cells, so that's why we know why is it there are interdigitations, so everything is here visible. Brush border of the proximal tubule cells is rich in one special enzyme. It is called alkaline phosphatase, which can be histochemically stained, and so if you do this proof of alkaline phosphatase, you can recognize this proximal tubules very quickly, but it isn't necessary. You will recognize them even without this proof, that according to all features which I already described. This is electron micrograph of the cell from proximal tubule. So microvilli on the apical surface, here is the detail of them. Many mitochondria, many lysosomes, and basal labyrinth. No nucleus is here visible because the cell is so big that it is easy to cut it at the level where nucleus isn't present. Detail of the basal labyrinth and the contact of the basal labyrinth and the basal lamina with the wall of fenestrated capillary. This is the endothelial cell, its lateral process of the fenestrated capillary. You can see diaphragma in a fenestration, and you can see that the basal lamina of this capillary doesn't fuse with the basal lamina of the tubule, so there isn't complex of basal laminae like we saw it in the renal corpuscle. And let's go to loop of Henle. As I already said, it has its descending and ascending limb, and each of them can contain, but not every time, thick and thin segment. I think that now it's good to say that there exists, sorry, I'll go to these pictures once more, but there exist two types of nephrons. About 1/6 of nephrons have got their renal corpuscles near the medulla. So they are called juxtamedullary nephrons. These juxtamedullary nephrons have got very long loops of Henle. Their loops have got quite short, thick descending segment, but very long thin segment, which goes down and up. So thin segment of these juxtamedullary nephrons, or loops of these juxtamedullary nephrons, have got descending and ascending segment. And then there is a quite long, thick ascending segment going to distal tubule. More nephrons are cortical. 5/6 of nephrons are cortical. The renal corpuscles are not so close to medulla, and their loops are much shorter. The loops don't go so deeply to renal pyramids. They go only to the outer parts of the medulla, and you can see that their thin segment is very short, and it is only in descending part, only here in the descending part of the loop, and the turn of loop and the whole ascending part is thick segment. So this loop hasn't got in fact ascending thin segment. And now these two skipped pictures. When we look at the medulla of the kidney, we can see that the arrangement of tubules there is much more regular. If you remember the pictures from the cortex, there were usually coiled tubules. So we saw in one place longitudinal section, cross sections, oblique sections. Here in the medulla, tubules go usually parallelly. And so we can see, for example, here only cross sections, or here only or almost only longitudinal sections of all tubules. And we can see thick segments of handless loop, which can usually be similar to distal tubules only when we are in the outer part of the loop. They can be like proximal tubule, because the descending thick segment of loop has the same structure as the proximal tubule. Ascending segment of loop has the same structure as distal tubule. Then we have got thin segments of a loop, like this or this. We don't recognize whether it is ascending or descending. They have the same morphology, but not the same function. The cells are there different, but we don't recognize it morphologically. Thin segments are lined with simple squamous epithelium. And the highest, the biggest, the largest things here, the largest tubules here are collecting ducts. Collecting ducts don't belong to nephron, so I'll describe them later. So in the medulla we can see thick segments of the handless loop, and we recognize if they are ascending or descending. Thin segments of handless loop where we don't recognize it, and collecting ducts, and also collecting tubules, but they are near the cortex, so not so deeply in the medulla. And this is the same in the longitudinal section. And but more here, and I didn't show it, there are also vessels, vasa recta. VR means vasa recta. If there is an erythrocyte inside, it's easy to recognize it, but if it isn't, they can sometimes resemble thin segments of loop. Let's say they are a little smaller. They are smaller. Thin segments measure about 30 micrometers. Vasa recta are smaller, but sometimes if they don't contain blood, the recognition is not so easy. You will see it in slide. And here it is easy because vasa recta have got a lot of erythrocytes, and according to the size of erythrocytes we can recognize their size. They measure like let's say 2 erythrocytes, 15 to 20 micrometers, so they are a little smaller than thin segments of loop. Thin segment of a handless loop is, for example, this here, or this here is a thin segment of a handless loop. On the other hand, thick segments of handless loop is, let's say, this, and this biggest thing is collecting duct again. Yeah. And this is electron microscopy. Description is clear. There isn't any thick segment of handless loop, so there are only thin segments of handless loop, and they call capillary, but we know they are called vasa recta. Okay, in this picture the size of them isn't so much different. And again, collecting tubules or ducts. High magnification of thin segment of handless loop, and thin segments of handless loop contain very squamous cells, very thin squamous cells containing only few organelles. That's a proof that the transport of ions and molecules through the cytoplasm of this epithelium is only passive. The descending parts, this is descending, this is yellow, is descending part of handless loop, of its thin segment, is permeable for both ions and water. Ions usually go in and water goes up, and the content, the urine in the loop, becomes more and more concentrated. Then is the turn, and the urine goes up, but the wall becomes permeable only to ions. And ions, when going up and when going to less concentrated interstitium, are resorbing back from urine, but water no. Water cannot go inside, and so the content becomes still more and more, the interstitium becomes more and more concentrated. And that's the principle how the renal interstitium becomes hypertonic. It is very important for the formation of hypertonic urine, which is then definitively concentrated while going through the collecting ducts. Collecting ducts are specialized by water reabsorption, and so urine is concentrated here, so the definitive urine is many times more concentrated than the primary urine. Each of us is producing about 1.5 liters of definitive urine per day. Compare it with 180 liters of primary urine, so it's the urine is more than 100 times concentrated. And we are back in the cortex. The loop is returning to the cortex and goes to distal tubule. Distal tubule measures about 40 micrometers in diameter, so it's smaller than the proximal tubule. So if you compare proximal and distal tubule, you recognize it easy. It's smaller. You will see well its lumen because there isn't any brush border. You can see again simple cuboidal epithelium, but the cells are much smaller, and so you can find many nuclei. Usually each cell has got its nucleus visible in our section. Compare it with this long segment of the wall of proximal tubule having no nucleus due to so big cells here. And also, if you compare the color of this distal tubule and this proximal tubule, I hope you will agree with me that distal tubule is darker, more eosinophilic. Eosinophilia is here in both tubules, but distal tubule, the cells of distal tubule don't contain so many mitochondria, so they are less eosinophilic. They are still eosinophilic, but less than cells of proximal tubule. So there are differences. The same is visible in longitudinal section. Proximal tubule, invisible lumen, big cell, high eosinophilia, higher diameter. Distal tubule, visible lumen, less eosinophilic cells, smaller diameter, more nuclei in the section. Electron microscopy of the distal tubule cell doesn't show brush border. It is important. The cells here have got some microvilli. You can see some short scattered microvilli, but they are not so many and not so long to form a brush border. Nevertheless, basal labyrinth, it is present. Basal labyrinth is present, as well developed, and so the nucleus is pushed a little above the central part of the cell. So basal labyrinth is present, but brush border no. The total amount of mitochondria is little smaller than in proximal tubule. Also the amount of lysosomes is not so high. Here mostly reabsorption of ions is present. Bigger molecules needing lysosomal digestions were absorbed already in the proximal tubule, so they aren't here. Ions transported by or reabsorbed by cells of distal tubule are directed, or this reabsorption is directed by a hormone, by a special hormone, aldosterone, belonging to mineralocorticoids produced by the adrenal cortex. So there are pictures showing the difference of proximal and distal tubule. The electron microscope can show it very well. I think this picture doesn't need any description. Everything is here written. More mitochondria, less mitochondria, no brush border here, brush border only here. Basal labyrinth in both tubules. They can be really in very close contact because they both are present in the cortex. But every time you'll find more sections of proximal tubule than distal tubule. It doesn't mean that the kidney contains more proximal tubules. Of course, their number has to be the same because each nephron has got one proximal and one distal tubule. But that's because they are convoluted, they are coiled, and the proximal tubule is longer. So proximal tubule can go more turns than distal tubule. Okay. When the distal tubule goes back to the cortex, it usually comes to very close vicinity of the vascular pole of this renal corpuscle, where originally the proximal tubule of the nephron began. This area where distal tubule is coming to the vicinity of vascular pole of the renal corpuscle is called, by this quite complicated term, juxtaglomerular apparatus. Juxtaglomerular apparatus is the structure having three components. In fact, it has a component which belongs to distal tubule. This is called macula densa. So one of the first, let's say the first component of the juxtaglomerular apparatus is macula densa, and it is a group of cells belonging to the distal tubule. The second component are juxtaglomerular cells. They are modified, specialized smooth muscle cells belonging to the wall of afferent and sometimes efferent arteriole. So juxtaglomerular cells are cells of the arteriole. And there are also some cells filling the spaces among two previous structures. They are called extraglomerular mesangial cells. They are similar to mesangial cells you already know from renal corpuscle. You can see that they have the same color in this schema. You know, these brown branch cells were in the corpuscle. We already know them. And the same cells can be found here in the area of juxtaglomerular apparatus, but it is not in the area of glomerulus. That's why they are called extraglomerular mesangial cells. Okay, so there are three components of juxtaglomerular apparatus. Why do we need juxtaglomerular apparatus? Juxtaglomerular apparatus is very important for the function of the kidney, because the cells of macula densa have a special chemoreceptors in its apical plasma membrane, and they can detect the amount of ions, mostly sodium ions, in the urine situated here in the distal tubule. This information about the sodium content is then given to juxtaglomerular cells. They receive this information, and if the amount of the sodium ions here is high, they secrete special enzyme. It isn't hormone, it is enzyme. It is called renin, special renin. It is a peptide which is secreted by juxtaglomerular cells to blood, and they stimulate conversion of angiotensinogen into angiotensine. It is another enzyme. This conversion is done in lungs, and angiotensine then has got two functions. It's directly influencing the smooth muscle cells, and it causes their contraction, and so the vascular diameter decreases, and the amount of blood going through the kidneys, but not only through the kidneys, it is the reaction of the whole body. But so the amount, the blood pressure is increasing, and the amount of blood going through kidney is decreasing. And the second target organ of the angiotensin is adrenal gland, and it stimulates production of aldosterone, which stimulates distal tubule to reabsorb sodium, and so the amount of sodium decreases in urine and increases in blood. And of course the opposite situation comes when the amount of sodium in urine is lower, then renin is not produced, and blood flow through kidney becomes faster and more voluminous. So let's look at morphology of juxtaglomerular apparatus. These extraglomerular mesangial cells can be sometimes also called Lassis cells, and they don't differ from the cells you know from corpuscle. Cells of macula densa are higher than the other cells of distal tubule. Normal cell of distal tubule is cuboidal, and here the cells are columnar, and they are tightly attached, tightly arranged in this area, which gives this area denser appearance. Mostly nuclei of the cells are more densely accumulated in this area. Juxtaglomerular cells have got small granules, but visible at the level of the electron microscopy only. I will show them, and they contain renin. This is macula densa. Look at the columnar cells with very densely arranged nuclei. Here is afferent arteriole, or probably this is afferent, can be also efferent arteriole, and some cell in its wall is labeled J, so it's juxtaglomerular cells, but it isn't easy to recognize it at this level when we don't see granules. And in between there are some extraglomerular or lassis cells. Okay, so we will try to find maculae densae in the slide. You will see mostly maculae densae, and the other parts of juxtaglomerular apparatus are not easy to recognize, but we know that they have to be near the macula densa. Again, typical arrangement of columnar cells in this part of distal tubules, so macula densa, lumen of an arteriole, so we can be sure that here must be some juxtaglomerular cells, and only when we use electron microscopy we can see electron dense granules containing renin in the cytoplasm of the juxtaglomerular cells. So this is the juxtaglomerular cells, or two juxtaglomerular cells. Here is macula densa and some mesangial cells here. So we can leave a nephron and we can go to system of collecting tubules and ducts. The collecting tubules are quite small. They have the same diameter as a distal tubule, that means about 40 micrometers. In some textbooks their beginning, their proximal portions are called connecting tubules and then collecting tubules, but I think it isn't so important. I would call all this system collecting. So collecting tubules, each of them comes from one nephron. They join together to form collecting ducts. Collecting ducts go at first through the medullary ray and then through medullary pyramid. They become thicker and thicker as they receive new collecting tubules from another nephron. So the biggest collecting ducts can measure 200 micrometers in diameter, and then they are called papillary ducts of Bellini, and they are going to the renal calyx. Here down is the tip of the renal papilla and renal calyx, minor calyx of course. The epithelium of all collecting tubule and ducts is simple. At the smallest tubules it's simple cuboidal. Then in collecting ducts and papillary ducts it's simple columnar. The cells of the tubules and ducts have typical apical protrusions, and so the cross-section of a lumen of such tubule or duct is like a star, and it's easy to recognize it in the slide. So star-like or stellate sections of the tubules. The cells of tubules are of two types: the much more frequent are so-called clear cells. They have not many organelles, but they have very many special proteins in the apical plasma membrane. These proteins are called aquaporins. The aquaporins, you already know them from the cytology. Aquaporins are special protein complexes allowing the transport of water, and they are sensitive. These aquaporins in the collecting tubules and ducts are sensitive for the ADH. It is a hormone, antidiuretic hormone, is a hormone from the posterior lobe of hypophysis, and it stimulates the reabsorption of water by aquaporin, and so the definitive concentration of urine is highly dependent on the level of antidiuretic hormone. If its level is high, the urine is concentrated, and if the level is low, for example, in case of the pituitary gland damage, we are not able to concentrate the urine, and the amount of urine per day is very high. The disease, this disease is called diabetes insipidus. So this is a small magnification of electron microscopy from some capillary tubule. You can see that its cells are still cuboidal, but they have their apical bulges, apical protrusion, giving the star shape to the lumen. And you can see that they are not very well equipped with organelles, so they are not able to actively transport ions or water. The transport here is passive but dependent on the function of aquaporins. And the lateral borders of these cells are straight. It was visible in the previous slide. There are no invaginations, interdigitations, and so we can see very well the borders in the section. That is another thing how we can recognize collecting tubules and ducts in the section. And you can see in this picture that there are not only these clear cells but also some darker cells, but it isn't easy to recognize them, but the level of electron microscopy can help us. Dark cells or dense cells of the collecting tubules and ducts are present here as well. They are lower, they are full of mitochondria, and we don't know many things about their function. Probably they are important mostly in ion transport, in the exchange of hydrogen and bicarbonate ions, and so the resulting pH, definitive pH of urine is dependent on the function of dark or dense cells of collecting tubules and ducts. And here are very high epithelial cells of the biggest papillary ducts. And the same in the electron microscope. Interstitial tissue of the kidney is loose connective, as I already said. It's very rich in capillaries. Capillaries are here fenestrated in the peritubular interstitium of cortex, and also in the medulla. Of course, in the medulla, capillaries are called vasa recta. Sometimes you'll find special interstitial cells here, and they are responsible for the secretion of another hormone. The kidney is a very potent organ in hormone secretion. Not only renin is here secreted, but also erythropoietin. interstitial cells secrete erythropoietin, which is a hormone stimulating erythropoiesis in bone marrow. And and we are leaving kidney, and we are going to urinary passages. Urinary passages are hollow organs, and so we can recognize some layers of their wall, as usually. And the first of them, the innermost layer of urinary passages is tunica mucosa. Tunica mucosa mostly consists of transitional epithelium, special type of epithelium typical for urinary passages only, and lamina propria mucosae. So this is the composition of tunica mucosa. Only urethra has got another epithelium than transitional, but the other parts of urinary passages have got transitional epithelium. Then the second layer is tunica muscularis, so there is no submucosa in urinary passages. Directly under the mucosa is tunica muscularis, and look at the orientation of its sublayers. It's very exceptional, and you should remember this. The first layer, the deeper layer of the muscularis in urinary passages, is longitudinally arranged, not circularly as usually, as you know from digestive tract, from the respiratory tract. Here is a longitudinal arrangement, and the outer layer is circular. Only in the distal part of the ureter there is the additional third layer or third sublayer of tunica muscularis, which is again longitudinal. But usually there are just two layers, longitudinal and circular. The outermost layer of urinary passages is usually tunica adventitia, because they are present either in the renal sinus, or ureter is present in the retroperitoneal space. Urethra is situated in the perineum, and so there is these organs are firmly connected to the surrounding connective tissue by tunica adventitia. Only urinary bladder is intraperitoneal organ, and so it has got serosa on the surface, having mesothelium on as a visceral peritoneum, in fact. Okay, so that's the general structure of urinary passages, and now let's look at the transitional epithelium. Transitional epithelium was described in the general histology in the epithelial tissue, so you already know that it belongs to stratified epithelia, which means that it's composed of several layers of cells, but it isn't possible to classify it according to shape of superficial cells, because its superficial cells are neither columnar nor squamous. They are very big, they are protruding to the lumen, they sometimes contain two nuclei, and they have got a process which is invisible in the light microscope, but I can draw it, which is going to the deeper layers of the epithelium. Originally it was described, or some time ago it was described, that this process is touching the basement membrane, and so this epithelium is in fact pseudostratified. But now we know that during differentiation these superficial cells lose their contact with the basement membrane. This process is not so long, so it's finished somewhere here, and it isn't so long to touch to reach the basement membrane here down. So now we classify this epithelium as a stratified type. Nevertheless, these superficial cells are still called umbrella cells because they really resemble umbrella with the holder. Under the umbrella cells there are several layers of intermediate cells, and their number is different the number depends on the distension of the organ. If some organ like ureter, this is probably ureter, when the ureter is contracted, empty, the amount of intermediate cells is very high. If ureter or bladder is dilated, there are just 2-3 layers of intermediate cells, and of course down are some basal cells which are undifferentiated and which divide and give rise to intermediate and at the end umbrella cells. The shape and arrangement of cells isn't the only special feature of transitional epithelium. They have very specialized apical plasma membrane. Plasma membrane on this apical surface of umbrella cells is very thick. It's first thing. It is very thick. It is almost impermeable. And it is important because inside is usually very highly concentrated urine here, which can contain many times more ions than cytoplasm of the cell, and if the membrane is permeable, the ions and water mostly ions would go in, and the cells would crash. So the membrane is composed of special mixture of phospholipids, where the most important is so-called cardiolipine, and this mixture gives the membrane very low permeability. The second thing are very special membrane proteins which are called uroplakines. They are differentiating only in these cells, so they are very special for umbrella cells. They accumulate under the membrane and they form thick plaques. It will be better visible in electron micrograph, or I use the schema. Plaques of uroplakines, and these plaques are important for formation of deep invaginations of the membrane, and these invaginations represent something like a depot of the membrane during the time when the organ is contracted, and they disappear when the organ is dilated, and then these plaques are situated on the surface. These uroplakines are connected with the network of intermediate filaments, and so they are responsible for the shape changes of the cells. So the apical plasma membrane and apical surface of the umbrella cells is really very special when you look at it in the electron microscope. They are usually very angular structures, some tips and right angles and invaginations, and sometimes these invaginations go on deeper to the cytoplasm, forming so-called fusiform vesicles, which are still composed of a membrane, of the superficial membrane, representing a depot of the membrane. And when the organ is dilated, and you know that urinary bladder can be sometimes very much dilated, these fusiform vesicles disappear, and the area of the apical plasma membrane becomes much higher, much wider. Yes, there are again fusiform vesicles and invaginations of plasma membrane, and in this high magnification we can see electron dense plaques of uroplakin. Or this is high magnification, so electron dense plaques of uroplakin under the membrane. Okay, let's go through the urinary passages. They are starting in the renal sinus as minor calyces. The transitional epithelium there is quite thin, mostly at the site of renal papilla. You can see papillary ducts going to the renal minor calyx. Papillary ducts developed by joining of collecting ducts, and this is the first place where some smooth muscle is situated in the wall. On the other side of the minor calyx, then is the renal papilla. Minor calyces join to major calyces, then to renal pelvis, which leaves the or then the urinary passages leaves the renal sinus as a ureter. This is the schema of the ureter wall, containing tunica mucosa, having epithelium and lamina propria. I didn't say, but it's here written that the transitional epithelium of urinary passages is sometimes also called urothelium. So if you read this this word in some textbook, don't be surprised. Urothelium, this is the same as transitional epithelium. Yeah, urothelium, lamina propria, and then two layers of smooth muscle. And as I already said, the innermost is more longitudinal, the outer is more circular. Of course, this longitudinal and circular appearance is in fact the result of the different spiralization. All muscles, every time in all tracts, are spirally organized, and their histological appearance depends on the height of the spiral turns. Near the urinary bladder then can be found the second longitudinal, so in total the third layer of smooth muscle tissue, and outside is tunica adventitia. And this is a section of a ureter. Typical star-shaped lumen, because the ureter is empty, of course, in the section, and so it's contracted. Transitional epithelium, lamina propria, and longitudinal smooth muscle, circular smooth muscle, together tunica muscularis, and adventitia. Tunica adventitia is outside. So no epithelium, no mesothelium on the surface. Now we are in the urinary bladder. The composition of a wall is similar to this we know already, so just to repeat: urothelium, lamina propria. Smooth muscle of urinary bladder is very thick, And as the organ is almost spherical, it's sometimes not so easy to distinguish longitudinal and circular arrangement of smooth muscle. Outside is serosa, as I already said. Serosa covered by mesothelium. Higher magnification, transitional epithelium, lamina propria, folds of the mucosa when the organ is empty, and also thick epithelium when organ is empty and contracted. Smooth muscle tissue forming many bundles, and serosa isn't in this picture, but you can imagine that it must be outside. Male urethra is much longer than female urethra, of course, and so it has got more parts. We are distinguishing 4 parts of male urethra, of urethra masculina: short pars intramuralis, then pars prostatica, which is going through the prostate, and both these pars intramuralis and pars prostatica, are lined with transitional epithelium. Then is pars diaphragmatica, or also membranacea, which is quite short. It is the place where the urethra is going through the sphincter, through the external sphincter, and the longest is pars spongiosa in penis. Pars diaphragmatica and pars spongiosa are lined with stratified columnar epithelium. Stratified columnar epithelium is not very frequently found epithelium, but it is here, and only a very distal portion of pars spongiosa, which is called fossa navicularis, is lined with stratified squamous epithelium, which is at first non-keratinized, but then on the outer surface of the penis becomes keratinized. So we can find 3 types of epithelium in the urethra: transitional epithelium, stratified columnar epithelium, and stratified squamous non-keratinized epithelium. The other parts of wall also depend on the place where is it. Everything is modified by prostatic glands, for example, but lamina propria usually contains some glands, or they are here, which are called paraurethral glands of Littre. Paraurethral glands of Littre are mucus producing glands in the lamina propria of the urethra. And when the epithelium is stratified columnar, we can find groups of mucus producing cells directly in the epithelium. They represent intraepithelial mucus glands, and they are called lacunae ureterales, or urethral lacunae of Morgan. Urethral lacunae of Morgan. They are intraepithelial glands. On the other hand, paraurethral glands of Littre are normal glands having secretory portion in the lamina propria and connected by duct with the lumen of urethra. Unfortunately, we don't see this duct here, but you can imagine that there must be in another part of the section a duct. Then there is some smooth muscle tissue, but it is usually connected with cavernous spaces of the penis, so we will describe the other layers of the male urethra in male reproductive system. Female urethra is much shorter and has got only two segments. Proximal, in fact, intramural segment is lined with urothelium again, but it's very short, and then the whole remaining female urethra is lined with stratified squamous epithelium. So there isn't any segment of stratified columnar epithelium. Glandulae parauretrales are here as well, but they are not litres but scanous glands, and they also secrete mucus. So morphologically they are the same as the parauretral glands in male. Okay, this is the last picture of this lecture, so thank you for your attention, and looking forward to you in the next lecture or in the practical training. So goodbye.